chat with you about their variety of science topics and studies that they do. Our next one is rapidly approaching next week. It's at Aeronaut Brewery in Somerville, and that's on Thursday, May 12th. So if you don't have either of these flyers, be sure to pick them up here. And he's also going to tell you about one more event we have coming up as well.
So the final event we're going to be talking about tonight is called DayCon. This is shorthand for our 3-day-long science conference. It's coming up on Saturday, June 11th. So it's an entire day of talks and workshops with graduate students, kind of an entire day of talks like this, all centering around, since this is an Olympic year, we chose the theme Bodies in Motion. So we'll be talking about different things like metabolism, to the physics of sport, to the genetics underlying athletic ability. And this year, we're really excited to be featuring the Museum of Science, who will be coming and doing a live eye dissection for us. So if that sounds at all interesting to you, or you really like El Pilon, we will give you lunch. Please register online at SITNboston.com/daycon.
Yeah, so without further ado, we'll let Mary take it away. Thanks, Mary.
Hey. Hi, everyone. Thank you, Steph and Katie, for that kind introduction. So I am a graduate student in Harvard's Biological and Biomedical Sciences program, and today I'm going to help you learn a little bit more about cardiovascular disease. So just to start, you may or may not be aware that cardiovascular disease is actually the #1 cause of death globally, and percentage-wise, it accounts for about 31% of death. And sometimes we think of cardiovascular disease as kind of like a Western problem, a problem of developed nations. But in fact, over 75% of these deaths actually occur in low or middle income countries. So what exactly is cardiovascular disease? So here we have a picture of the body, the heart, and then what I like to think of as kind of the superhighways of the body are blood vessels, veins, and arteries. And today I'm going to focus more on the vascular, on the vessel side. But cardiovascular disease can really encompass so how do we define cardiovascular disease? We can look at the percent of the artery that's blocked by this plaque. So we can define this as the percent occlusion. And you can actually image this. It's pretty cool. You can inject a tracer into someone and then follow the blood through their vessels. And here you can see where the blood path narrows. That indicates that the artery is occluded because the blood is taking a narrower path. And so you can actually calculate and say, oh, this artery is 50% or 60% occluded. We can also define cardiovascular events. So for example, heart attack or stroke, something cardiovascular that lands you in the hospital. Or also people who have procedures to open up their arteries, perhaps to prevent further events. So a stent, which just opens up the artery, or a bypass, which just like a road, gives you a way around a blockage. And atherosclerosis is really not new. I found this study, they looked at 4 different pre-industrial populations. So ancient Alaskans, southwestern population, southwestern United States, Peru, and ancient Egypt. And they were actually able to find evidence of atherosclerosis in all 4 of these populations. So I hope by now I've convinced you that this is a human problem. And it's exacerbated by our current diet and lifestyle. So when we think of risk factors for cardiovascular disease, smoking is a big one. Smoking damages the vessels. High blood pressure, also very damaging to the vessels. Diabetes and obesity, which is actually what my lab studies. And increased age. This disease really develops over time. We can also think of cholesterol abnormalities as playing a big part in cardiovascular disease. You may have heard of HDL, which is our good cholesterol, and LDL, which we think of as our bad cholesterol. So if you have alterations in the amount of cholesterol in HDL or LDL, that can change your disease risk. And one thing I would like to point out is that these are called good or bad cholesterol, but they're actually, not sure why. Can everyone hear me or should I? Okay, great. So these are actually what we call lipoprotein particles. Don't be intimidated by the name. It just means there's cholesterol and fat and protein. So these are not just cholesterol. And we'll see that coming up. So after that quick introduction, here's our roadmap for the evening. So I'll give you a brief introduction to cholesterol metabolism and how that relates to cardiovascular disease. We'll talk about our standard therapy, which is statins. And then we'll take a quick break and we'll talk about new therapies developed from human genetics research. And I couldn't talk about cardiovascular disease without talking about nutrition. So we'll dive into dietary fats. Okay. So first I'd like to start with some cholesterol myths. And one is really that cholesterol is just bad. This is patently not true. Cholesterol has a lot of useful functions in the body. And this is what cholesterol looks like if you're a biologist or a chemist. Some of you may recognize these structures are carbons here, all bonded together. But the important thing here to see is that cholesterol has a unique shape right here, these rings kind of bound together. And this shape comes up in other structures as well. It comes up in our sex hormones, in estrogen and testosterone. They have the same four-ring structure. And actually both of these compounds, as well as progesterone, are made from cholesterol. So we need cholesterol in order to make these. Another picture you might remember from high school biology, this is our cell membrane, the phospholipid bilayer. So here are phospholipids, and there are two of them on each side. And what you can also see is there's some yellow here, and this is actually cholesterol in the membrane. And cholesterol is able to give these membranes some extra support. It's a vital structural component of every cell in the body. Another myth I hear a lot is that cholesterol only comes from your diet. Again, this is false. So here's your liver, and your liver is the main source of cholesterol in your body. It makes most of it, although there are other organs that can also make cholesterol. Okay, so I said the liver makes cholesterol, and it will package it for release into particles called VLDL, or very low density lipoprotein. And so you can see here this particle contains our protein ApoB, contains fats, and it contains cholesterol. And this is almost just like mailing a package. The liver is going to package these particles, and they can move all over the body. And VLDL has an especially important role in that it delivers fats to many of our organs. And fats are a really good source of energy, especially for organs like skeletal muscle that have a really large energy demand. So once the fat is removed from VLDL, it becomes LDL, our low density lipoprotein. And there may still be a little fat left in this particle, but mostly it's cholesterol rich. And these particles can be cleared by a protein on the liver called the LDL receptor. So they'll bind, and then the receptor will go into the liver cell and bring the cholesterol into the liver. The trouble is that this process is really not very efficient, and so we end up with a lot of LDL that stays in the blood. And as you may have guessed from the fact that we call LDL bad cholesterol, this is something that can lead to cardiovascular disease. for the spring lecture series, you may recognize this is from Vinny's talk, The Immune System in the Skin. And then my May the 4th reference is Joy's Storm. What we end up with is a complex plaque. And so our plaques have all kinds of immune cells. So all of these different types of immune cells here, they have a lot of dead cells, as I said, and they reside below this endothelial layer, below the cells lining the blood vessel. But what's really dangerous is when the plaques become unstable enough to rupture through the endothelial layer and move into the circulatory system, into the blood vessels, where they can potentially cause blood clots. And again, just to hit this point home that this is a disease that happens over time, it's very much a progressive disease. You don't wake up one morning and have cardiovascular disease. This is a process that starts very, very early in life. You'll start to have cholesterol accumulation on the vessels, and then it'll build up. Okay. So it'll build up over time as the plaques become larger and larger. Okay. Luckily, we do have a way to prevent plaque, and this is HDL, or good cholesterol. And HDL is a different type of particle than LDL. The two cannot interconvert. But what HDL can do is it can take cholesterol out of LDL, out of macrophages, and even out of plaques, and then bring it back to the liver where it can be disposed of. And lifestyle affects HDL, and generally things that we think of as healthy will raise HDL. So for example, exercise, eating plant foods, so fruits and vegetables that are high in fiber, and then also the consumption of unsaturated fats, such as those found in olive oil. So now I've given you a quick introduction to cardiovascular disease, and just to recap, we've shown that LDL in blood promotes atherosclerosis, but really these plaques are a mixture of cholesterol and immune cells, that the plaques arise from an immune response. But luckily, we do have our good cholesterol particles, our HDL, that can bring cholesterol back to the liver. And so we can pause for questions. Yes?
What are triglycerides and how do they relate to HDL?
Yes. So the question is, what are triglycerides? How do they relate to HDL and LDL? So triglycerides is the more scientific term for fats. So when I showed VLDL, that's bringing fats into the circulation, that's the VLDL is packaged triglyceride and cholesterol. The other thing you might be getting at is that some people think that excuse me, so the composition of triglycerides, the composition of fats in your diet plays a role in cardiovascular disease risk, and that's something we'll talk about a little bit later. Does that answer your question? Yes.
How do the omega-3 fats balance out the omega-6 fats?
Yeah. So the question is about omega-3 and omega-6 fatty acids and how they balance each other out. So just to give a little bit of background to that question, different fats have different properties, and omega-3s are thought to be sort of beneficial, anti-inflammatory, whereas omega-6s are thought to be more inflammatory and more negative. I will say that this is not very well understood. So One of the reasons why we think omega-3 fatty acids are so good is because of epidemiological studies. So studies of people over time that link but it's a pathway that's thought to be very beneficial in the body. And I can go into a little more detail after the talk if you'd like. Okay, yes?
Beyond the HDL, is there any other way to undo the plaque? What do we have?
Yeah, so the question is about non-HDL ways to undo the plaque. So in terms of natural, like other plaque busting molecules in the body, I'm not aware of any. And then there's a lot of interest in kind of synthetic HDL that we could administer to people to break up plaques. But this research is in very early stages. Yeah. Okay, so those are all great questions. And so now we can move on to our next topic, which that are a little bit more diverse. So these are the Omni cohorts. And the Framingham Heart Study helps us measure and evaluate someone's cardiovascular disease risk. So you can actually go to a website from the National Heart, Lung, and Blood Institute. You can put in a few key parameters. So I just did this for a hypothetical 50s male with high cholesterol. And it'll spit out a risk score in a percentage. So this hypothetical person's risk score is 10%, which means that according to the Framingham Heart Study, 10% of people who have this matching profile, this blood pressure, this cholesterol, will have a heart attack in the span of 10 years. And a 10% risk, we would definitely want to treat this person to try to prevent cardiovascular disease. So looking back at our cardiovascular risk factors, one really stands out in terms of what people have manipulated so far in treating cardiovascular disease, and that's our LDL, our bad cholesterol. So to lower LDL, we now have statin medications. You may be familiar with these brand names, Zocor, Lipitor, Crestor, or the generics that all end in statin. And during the time when Lipitor was under patent protection, it actually became the most prescribed drug in history, had over $125 billion in sales, and each year it accounted for about a quarter of Pfizer's total revenue. So these were really, really blockbuster drugs, and now as generics, they continue to be very, very widely prescribed. So what do they do? So statins actually inhibit the synthesis of cholesterol in the liver. So you may be wondering how they can do this. I told you that every cell in the body needs cholesterol. How can we just block the liver from making cholesterol? So in fact, this is actually the key to statins. When you block cholesterol synthesis, you make the liver cholesterol deprived. The liver has to figure out another way to get this cholesterol. And to do this, it actually is going to increase the level of LDL receptor on the cell's surface, and thus it's going to pull more LDL out of blood, break down those particles, and take the cholesterol from them. So this is how statins lower LDL. In addition, statins have another beneficial property in that they reduce inflammation. So even in people who have plaque, you can reduce the inflammation and potentially make these plaques less dangerous. And we really like statins to do two jobs. We want them to, first of all, do primary prevention. And what this is preventing a cardiovascular event in someone who's never had one before. We'd also like them to work for secondary prevention, so preventing another event in someone who's already had one. And it turns out that statins work really well for secondary prevention, but they don't necessarily work as well for primary prevention. So it depends on your risk level. This is one of the reasons why we don't recommend that everyone take a statin. If your risk is very low, statins will likely not be helpful. If your risk is around 7.5% to 10% or even higher, based on the Framingham Heart Study, your doctor will likely prescribe a statin. So despite these being blockbuster drugs, they're not perfect. There are a few problems. So one is that for statins to work, we need them to increase the level of LDL receptor on the surface of the liver cells. We need them to be able to other genetic variants that would indicate how well someone might respond to statins. I'm actually going to hold that question because we'll talk about it a little bit in the next section. I will say there are some people, so for example, it's rare, but if you don't have the LDL receptor, obviously you wouldn't be able to take a statin, but we'll look at that a little more closely coming up. Yes?
Is there a test for the person that can't tolerate the LDL as a receptor, or they just have to try it, then if they do a blood test and see if it doesn't work?
So for someone who doesn't have the LDL receptor, it'll be pretty obvious in their blood workup. Yeah, so, and again, I'll go into that a little more in the next section. And as we move kind of into this genetic and personalized medicine era, there may end up being more sequencing of people for these kind of harmful variants. It's not very commonplace in the clinic right now. Yes?
Do the statins actually destroy the plaque?
Yes, so do statins actually destroy plaque? That's a good question. So from what I read, I believe it's more of a stabilizing effect. I don't think it's so much that they destroy the plaque, but I'm not an expert in that area. So, but again, it's not as if you have to destroy plaque to decrease your cardiovascular risk. You know, if you do have kind of stabilized plaques just below that cell layer, you know, and they're not rupturing and they're not completely blocking blood flow through the artery, you may very well not have a cardiovascular event. So it's kind of, it's shades of gray, really. Okay, great. So with that, we can move to our intermission. And you may have heard that sitting and a sedentary lifestyle increase your risk of cardiovascular disease. It's been in the news a lot lately. So take this time to stand up and move around a little bit, and then we'll come back soon.
We'll take like 10 minutes. The bathrooms are up here. There's some more snacks. And also we have our survey up front. So if you have a few moments and want to start filling that out for Mary, that would be great. I thought the mic wasn't on it.
Now I feel a little bit louder. I moved it up a little bit. Yeah.
Like, do you think the mic was cutting in and out?
No, I think it was. Was there problems with the live stream?
So the live stream was dropping, but that shouldn't have anything to do with my.
I think it was just I had it a little bit further down. So
Yeah.
I can maybe I'll
All right. Let's just get
let it stay.
All right, we're gonna get started again. Thanks, Mary.
Okay, great. So now we're back for the third section of the talk. It seems like the mic is working a little bit better as well. So now I'm going to talk to you about human genetics. We had a question earlier about genetic variants, and so we'll be talking about that a little bit coming up. So I've talked about cardiovascular disease risk, but I want to make the point here that this is what we call a complex disease. And complex diseases are those in which both genetics and environment play a role. You can see we have a number of different DNA strands here. And this is to represent that there are many, many genes that play a role in your cardiovascular disease risk. And the question researchers are asking now is if we can look to human genetics and find, first of all, if we can better understand this disease, and secondly, maybe if we can find new options to treat it. So I've said many genes play a role, but there are actually rare cases in which a single gene has a large effect, and that's where our story starts tonight. So you remember our familiar LDL receptor that's going to clear our LDL out of the blood, bring it into the liver. And so as I alluded to in an earlier question, if you have a mutation in the LDL receptor such that it no longer clears LDL out of the blood, this will basically increase your risk of heart disease or cardiovascular disease. Likewise, you may have a mutation in a gene ApoB, which we know is our protein component. To 1,000 milligrams per deciliter, very, very high. In fact, so high that you can even get cholesterol deposits in your tissues. So this here is a cholesterol deposit, what we call a xanthoma, and these can form really all over the body. And so in 2003, scientists actually found another gene that can cause familial hypercholesterolemia, and this gene is Pcsk9. Don't worry about what it stands for. The name is really not useful for our purposes. But what we need to know is that when Pcsk9 is overactive, they found that it increases cardiovascular disease risk. And so they wondered in other people, in people who have a less severe cardiovascular disease risk, could Pcsk9 be a good drug target? If we blocked Pcsk9 in these people, would it prevent cardiovascular disease? And so they actually went and looked for people who naturally had less Pcsk9 activity. So Pcsk9 doesn't work as well. And they found, in fact, that if you have a mutation where you have inactive Pcsk9, can't do its job, you decrease cardiovascular disease risk. And in fact, there are 2 people in the world so far that they've found who have no Pcsk9 at all. Their LDL is less than 15 milligrams per deciliter compared to what we think of as really good is about 100, 110, 130 is pretty good. So this was a pretty big finding. This was pretty exciting for the cardiovascular field. So I've told you about this protein that has a lot of initials. What does it actually do? And so it turns out that it plays a role in LDL receptor recycling. So I showed you before that the LDL receptor brings LDL into the liver. What I didn't show you is that the receptor will actually go back to the cell surface so that it can take up more LDL and just continue this cycle. Turns out PCSK9 actually inhibits this process, it blocks it. So when PCSK9 binds the LDL receptor, when the receptor goes into the cell, it's broken down, it won't come back out again. And so when you have more PCSK9, your level of LDL receptor on the surface of the liver cells is lower. And actually, it's interesting because PCSK9 may account for some statin failures. So we said that when we treat with statins, we want the liver to increase the level of LDL receptor. It turns out that PCSK9 levels also increase, and this is variable and increases in some individuals more than others. So in people whose PCSK9 is very, very high, it's going to get rid of the LDL receptor on the surface of the liver cells. So it basically negates the positive effects of statins. And PCSK9 is really a unicorn within the biomedical field. So the field discovered in 2003 that a mutation in this gene could cause familial hypercholesterolemia. In 2015, there was already a drug approved to block this protein. So 12 years. This is incredibly fast in biomedical science. This really basically unprecedented. So Pcsk9 inhibitors, what do? So they're going to block the ability of PCSK9 to bind to the LDL receptor, and as a result, there will be more LDL receptor on the surface of the liver cells. And thus, the liver will pull more LDL out of the blood. And so this is similar to statins, except that these drugs work better. And so you can actually compare, this is data from a trial. They had people either on statins, our standard therapy, or they gave them statins and a PCSK9 inhibitor. And you can see that statins keep our LDL cholesterol right around 100. When you add a PCSK9 inhibitor, you decrease that by over 50%. And so the question here really is, we're lowering LDL, but do these drugs really improve cardiovascular disease? Do they improve health? And are there negative side effects? And the answer to both of these questions is really that only time will tell. So I don't think I need to remind you that our overall goal is not just to reduce so these are injectable drugs right now, and they cost about $14,000 a year. This is compared to statin pills, which are now generic. You take one in the morning and you're done. And so a lot of people, a lot of insurance companies and physicians are wrestling with the question of really who should take these drugs. So people at a high risk for cardiovascular disease, most likely, but it's a difficult balance. You know, how much better do these drugs have to be to justify this higher cost? And at this point, there are no clear answers, but hopefully once we know more about how these drugs actually hopefully reduce cardiovascular risk, we can begin to better answer these. So just to summarize part 3, I hope I've convinced you that PCSK9 is really a success story for biomedical research and human genetics in particular. And so far that they appear to really be powerful drugs against cardiovascular disease, but we need longer term studies to really determine how effective they are. And so I can take questions at this section. Yes.
You mentioned two people genetic cultures.
Yeah, so there are two people in the world who have these. Yeah, so it's very, very rare to have both copies of Pcsk9 not work. It's very, very rare. But these Pcsk9 inactivating mutations, they seem to be more common in African populations. Did you? Yes, so actually one of them is a yoga instructor. And they discovered I think she dislocated her shoulder or something, and she ended up at the emergency room, and they ran a lipid panel, and they ran it again, and they realized that, she was, more than a genetic 1 in a million. Yes. Yeah. So they've run really a battery of tests on these people, and I should say that's another thing that I made drug companies confident in targeting Pcsk9. If you can show that in humans there's no negative effects of getting rid of something, that's a strong rationale that maybe this drug might not have bad side effects. So Pcsk9 in animals or cardiovascular disease in animals in general? Yeah, so there has been a lot of animal research in Pcsk9. So they've done lots of studies in mice, and that was how they originally showed that this protein binds the LDL receptor. So it's very interesting. Probably the ideal animal to do cardiovascular disease research in for us would either be non-human primates or actually pigs. Pigs have very similar cholesterol metabolism to humans. So I work in mice. Unfortunately, mice are actually very well protected against atherosclerosis. So it's not an ideal model for this sort of research, but they're much smaller animals than pigs, and they're much easier to work with. No. Although I will say there was a therapy that was developed to try to make us more like mice, to get rid of a protein that mice don't have, because they thought it might improve our cardiovascular health. The protein is called CETP, if anyone wants to look it up. Yes?
One question I have, you put up a statistic about, so the risk of having a certain fad was about, so one copy was about 1 to 500.
Yes.
And then you said the chance of having both copies bad from both parents makes you like 1 in a million. I didn't understand. Is that just a rough number, or it seemed like it was?
So they're exactly what they sound like. They're these little droplets of cholesterol and fat. So that's a good question. So you can also, yes, so there is also our most well-known method for cholesterol excretion, like the defined pathway for getting rid of cholesterol, is bile. So you can convert cholesterol into bile, and then some of that is lost in the feces. Did you have another question? No. Yes.
So just to make sure I understand the terminology right, so there is a gene that expresses PCSK9, which is a protein.
Yes.
And more PCSK9 is a bad thing.
Yes.
Right? So more PCSK9 is a bad thing for cardiovascular.
Yes.
Is PCSK9 helpful for anything else?
Yeah, that's kind of the $1,000,000 question. Is there anything good about PCSK9? And unfortunately, there's no answer yet. But it's an evolutionary question. Why would this protein even exist if it has no beneficial function?
So an unrelated question: does fatty liver have anything to do with storage of all this cholesterol?
Right. So fatty liver, does that have to do with cholesterol? We tend to think of fatty liver more in terms of triglycerides, especially since triglycerides, there are different types of fat, basically. So triglycerides are relatively stable within cells, but there are also fats that we call reactive. And so these fats can do things, they can signal and send messages in the liver in ways that are detrimental to health. And so these reactive species are bad. And then also if you have too much triglyceride, too much fat in the liver, you can compromise liver function. And eventually you can also end up with cirrhosis, which is more severe liver damage. But we don't typically think of that as cholesterol mediated. It's more fat. Yes.
I'm told that we actually generate fat from the starches more so.
Yes.
Than food.
Yes. So that's a good question. So the body doesn't want to keep too much sugar in the blood. So we're very good at converting it to fat and sticking it in the liver. And so this is one of the reasons why people think that our diets that are high in processed sugars are promoting fatty liver. That's not to say that sugar doesn't have a purpose. So for example, your brain can only run on sugar. It can't run on anything else. But in the quantities that we consume sugar and starches, they can have really negative effects. And I think, was there a second part to that question as well that I missed?
To clarify, the starches do get turned into sugars, and sugars get turned into fats.
Yes.
And do you know what kind of fats they get turned into? Triglycerides, LDH, HDH?
So typically we think of them being turned into triglycerides, but there are other lipids as well.
Is that where the omega-3s and the omega-6s come in and balance things out or something? Or they don't know?
We can talk about it a little more after. It's a complicated picture. I will say that I think the interplay of different nutrients and different molecules made by the body is much more complicated than anyone might have envisioned, even just 40 or 50 years ago. Okay, great. So again, really great questions. You guys are very well informed. And now I'm going to talk about dietary fat, and I'm sure we'll have lots more questions. So to start really with the very basics, so we'll start with our fatty acids, and these make up our triglycerides. So this is a picture of a saturated fatty acid. And again, if you remember your horror stories from organic chemistry, these are all carbons, and they're connected with single lines. So these are single bonds. Here we have an unsaturated fatty acid, which looks a little bit different. So it has these double line here. connecting the carbons. And so any fatty acid that has at least one double bond is unsaturated. And we think of saturated fats, we tend to think of them as coming from animal products. They're solid at room temperature and they're associated with cardiovascular disease. Whereas our unsaturated fats are liquid at room temperature, and we typically think of them as being healthy fats like vegetable oils. And I actually didn't know until a couple years ago how we got the idea that saturated fats were bad for cardiovascular health. So I'll show you that data now. This is from a work by Ansel Keys. It's called the 7-Country Study, and it was undertaken in the 1960s. And Ansel Keys was really interested in understanding, like the researchers in the Framingham Heart Study, really what was causing cardiovascular disease. And here he plotted the percent calories from saturated fat, so how much saturated fat is in the diet, versus the cardiovascular events or death per 100 people. So seeing how much saturated fat a population ate versus how much cardiovascular disease they had. And so he plotted this for a number of different populations, each here shown with a circle. I think the US is actually this one right here. And what you might notice is that you can draw a line here and you can say that saturated fat is correlated with cardiovascular disease. So you can't say that saturated fat causes cardiovascular disease, but you can say that they might be related. And this actually spurred some research into replacing saturated fats. And it led us to develop margarine. So for margarine, they take unsaturated fatty acids, so here, our double bond here, and what you can actually do is partially hydrogenate them. Don't really worry too much about what that means, but basically you're changing the structure of the fat. So now this is a trans fatty acid, and except for this one double bond here, it looks a lot more like a saturated fat. And so people thought, okay, these are not saturated fats, maybe they'll be healthier. They also had the additional benefits of being cheap and easy to transport and having a long shelf life, so you could put them in processed foods and not worry about the food spoiling. And the question researchers started to ask at this point is if these trans fats were healthier than saturated fats. And now we have the benefits of living in the 21st century. We already know the answer to this question, but we'll walk through why this is. So I told you that HDL can take cholesterol out of LDL and bring it back to the liver. But there's actually also a process that goes the other way. Cholesterol goes from HDL to LDL. And it turns out that trans fats actually increase this process. So trans fats give us what we call the cholesterol double whammy. So they increase LDL, our bad cholesterol, and they decrease HDL, our good cholesterol. As if that weren't bad enough, they also increase inflammation, and they increase dysfunction of this endothelial, this single cell layer. So I'm really not understating it when I say these are a cardiovascular nightmare. And in fact, there's no debate about trans fats. There are a lot of debates in nutrition right now, and this is really not one of them. So the FDA in 2015 finally took this generally recognized as safe designation away from trans fats and said, these have no health benefits, there's no reason to have these in food anymore. And they gave manufacturers 3 years to completely remove them from food. The good news is that other work by the FDA to label trans fats actually decreased trans fat consumption in the US by about 78%. So what do we predict will happen if we don't have any artificial trans fats? So a 2008 estimate estimated that we'd save 50,000 lives per year. So this is about 8% of the total deaths from cardiovascular disease in the US. But what if we just reduce that little residual trans fat consumption that we still have? They estimate that we could, again, reduce about 1% of deaths, about 3% of heart attacks. This may not sound like a lot, But if we can just get rid of this one thing that doesn't need to be in our food to begin with, you know, we can really save lives. So what about saturated fat? You've probably read and heard a lot about saturated fat in the news. And one of the reasons why saturated fat gets a bad rap is because it raises our LDL cholesterol. So this goes back to what you were talking about earlier in the question period. The problem is that saturated fat is not quite as simple as we might want it to be. So we have to look at what happens when we replace saturated fat with other kinds of nutrients. So if we replace saturated fat with trans fat, this is obviously going to be a bad change because we know how horrible eating whole, minimally processed food that is as close to its natural form as possible. So eat food, not too much, mostly plants, as we've all heard. And again, I hope, as hard as it is, that in the future we can realize that food quality is really what's important, and that our nutrition label really doesn't tell us everything. Just because two things have the same number of calories doesn't mean that they have the same metabolic effects. So to summarize, we've gone through a lot of the basics of cardiovascular disease. And so if you can take away these 4 points, that would be great. The first being that cardiovascular disease is really very complex genetically and metabolically. So we see it develops over time. There's inflammation, cholesterol. There are a lot of things at play. And that statins, our most successful cardiovascular therapy so far, increase the uptake of LDL by the liver. And Pcsk9 inhibitors appear to do that even better. And also, I hope I've convinced you that, you know, nutrition and public health can make a difference, and that, you know, you can affect your environment and affect your cardiovascular risk. So thank you very much for attending, despite the rainy weather, and I'm happy to take a few more questions. Yes.
One thing you had mentioned, but not get too much in detail. How about the what we call lifestyle changes? Beside drugs and so on, and sometimes it's kind of confusing. The thing about drugs, you have dosage, but you are wrong. Lifestyle changes. You see, here good examples, but
Yeah.
it is quote unquote almost free. How come it's not the most effective thing to do?
Okay, so I'm not entirely sure. Do you want me to talk more about specific lifestyle changes a person can make to reduce cardiovascular risk?
Because, you know, you have developed some of the risk factors we do not control well, like aging. We cannot
Unfortunately, no.
You know, the other thing that people talk about a lot,
Yeah.
you talk about the dietary changes.
Yeah.
But how much you eat depends on what
Right.
your lifestyle is.
Yeah. So the
Right? There are a lot of other, there are so many lifestyle things, and it's almost confusing. How many can you do?
Yeah. Right.
Do only one.
So I mean, I'm not a cardiologist, but I think the medical establishment really wants you to focus on changes that you can actually continue. You know, just cutting out salt for a month and then going back to a high salt diet isn't very effective. So I think, you know, not smoking is really helpful for cardiovascular health. again, if you can decrease your salt intake, which will generally, if you have high blood pressure, will help manage that. A number of people also take medications for high blood pressure. If you can eat more fiber, exercise, those things I talked about with raising HDL. I think those are tangible things that a lot of people can do. You may not eat salads every day, but you can, add a few more vegetables to your dinner plate. These kinds of small changes. Also, weight loss can be helpful. Small amounts of weight loss.
Also, public policy lifestyle change can be designed
Right.
to people's life, their income,
Right.
and and
Have higher soda taxes and such. I hope that answers your question. We can always talk more after. Yes.
So you'll have some more carrots next time instead of just cookies out there?
Yes, I will say I did not pick the snacks. But yes. Yes.
That Framingham heart study, is that due to go on forever? I don't think.
That's a good question. As far as I know, it's really kind of going strong. It's a mainstay program. I'm not sure when and if they'd recruit a fourth cohort, the great grandchildren of Framingham. But again, it's produced such useful insights that I would see it continuing. Yes.
Also, they're going to do genetic studies on some of the participants of the Framingham studies.
Yeah, so genetics in the Framingham study, that I'm not entirely sure about, but that is a new trend in the field to basically look at how genetics alter LDL, HDL, frequency of cardiovascular disease. So human genetics and cardiovascular disease is really an exploding new field. Yes.
On many food labels, like the one you showed on the previous slide, they have a line with cholesterol.
Mm-hmm.
That's not exactly.
Right. Yeah. Okay. So this is something I didn't talk too much about. So I said the body can make cholesterol. We do also get cholesterol from our food. These labels can be a little misleading because your body doesn't necessarily absorb all the cholesterol that's in food. So some of it is actually lost in your feces without ever entering your system, really. And current research suggests that the amount of cholesterol in your diet may have some impact on your cardiovascular disease risk, but the amount of cholesterol your body makes naturally seems to have more impact.
So that's already in food.
So yes, so this is a hypothetical nutrition label.
versions of them there. Feel free to look there and
Yes.
download them.
Yeah, and one other thing I should say is that a lot of these images are actually from an article I wrote for Science in the News' blog about PCSK9 inhibitors, and there are some links on your info sheet for articles there.